Hydras undergo morphallaxis, tissue regeneration, when injured or severed. Typically, hydras reproduce by just budding off a whole new individual. The bud occurs around 2/3 of the way down the body axis. When a hydra is cut in half, each half regenerates and forms into a small hydra. The head regenerates a foot and the foot regenerates a head. This regeneration occurs without cell division. If the hydra is sliced into many segments, the middle slices form both a head and a foot. Two pairs of positional value gradients explain the polarity of the regeneration. There is both a head and foot activation and inhibition gradient. The head activation and inhibition work opposite to the pair of foot gradients. Grafting experiments showed evidence of these gradients in the early 1900s. The inhibitors for both gradients are important to block the bud formation. The location where the bud forms is where the gradients are low for both the head and foot. Hydras are capable of regenerating from pieces of tissue from the body and additionally after tissue dissociation from reaggregates. This process takes place not only in the pieces of tissue excised from the body column, but also from reaggregates of dissociated single cells. It was found that in these aggregates, cells initially distributed randomly undergo sorting and form two epithelial cell layers, in which the endodermal epithelial cells play more active roles in the process. Active mobility of these endodermal epithelial cells forms two layers in both the reaggregate and the regenerating tip of the excised tissue. As these two layers are established, a patterning process takes place to form heads and feet. Non-senescence. Daniel Martinez claimed in a 1998 article in Experimental Gerontology that Hydra is biologically immortal. This publication has been widely cited as evidence that Hydra does not senesce, do not age, and that they are proof of the existence of non-senescing organisms generally. In 2010, Preston Estep published, also in Experimental Gerontology, a letter to the editor arguing that the Martinez data refutes the hypothesis that Hydra does not sense. The controversial unlimited lifespan of Hydra has attracted much attention from scientists. Research today appears to confirm Martinez's study. 26. Hydra stem cells have a capacity for indefinite self-renewal. The transcription factor, forkhead FOXO, FOXO, has been identified as a critical driver of the continuous self-renewal of Hydra. 26. In experiments, a drastically reduced population growth resulted from FOXO downregulation. 26. In bilaterally symmetrical organisms, bilateria, the transcription factor FOXO affects stress response, lifespan, and increase in stem cells. If this transcription factor is knocked down in bilaterian model organisms, such as fruit flies and nematodes, their lifespan is significantly decreased. In experiments on H. vulgaris, a radially symmetrical member of phylum Cnidaria, when FOXO levels were decreased, there was a negative effect on many key features of the Hydra, but no death was observed. Thus, it is believed other factors may contribute to the apparent lack of aging in these creatures. 5. DNA repair. Hydra is capable of two types of DNA repair, nucleotide excision repair and base excision repair. 27. The repair pathways facilitate DNA replication by removing DNA damage. Their identification in Hydra was based, in part, on the presence in its genome of genes homologous to ones present in other genetically well-studied species playing key roles in these DNA repair pathways. 27. Genomics. An ortholog comparison analysis done within the last decade, as of, demonstrated that Hydra shares a minimum of 6,071 genes with humans. Hydra is becoming an increasingly better model system as more genetic approaches become available. 5. Transgenic Hydra have become attractive model organisms to study the evolution of immunity. 28. A draft of the genome of Hydra magnipopulata was reported in 2010. 29. The genomes of cnidarians are usually less than 500 megabases, in size, as in the Hydra viridissima, which has a genome size of approximately 300 megabits. In contrast, the genomes of brown hydras are approximately 1 gigabit in size. This is because the brown hydra genome is the result of an expansion event involving lines, a type of transposable element, in particular a single family of the CR1 class. This expansion is unique to this subgroup of the genus Hydra and is absent in the green Hydra, which has a repeating landscape similar to other cnidarians. These genome characteristics make Hydra attractive for studies of transposon-driven speciations and genome expansions. 30. Due to the simplicity of their life cycle, when compared to other hydrazoans, hydras have lost many genes that correspond to cell types or metabolic pathways of which the ancestral function is still unknown. Hydra genome shows a preference towards proximal promoters. Thanks to this feature, many reporter cell lines have been created with regions around 500 to 2,000 bases upstream of the gene of interest. Its cis-regulatory elements, CRE, are mostly located less than 2,000 base pairs upstream from the closest transcription initiation site, but there are CREs located further away. Its chromatin has a RABL configuration. There are interactions between the centromeres of different chromosomes and the centromeres and telomeres of the same chromosome. It presents a great number of inter-centromeric interactions when compared to other cnidarians, probably due to the loss of multiple subunits of condensin II. It is organized in domains that span dozens to hundreds of megabases, containing epigenetically co-regulated genes and flanked by boundaries located within heterochromatin. Transcriptomics. Different Hydra cell types express gene families of different evolutionary ages. Progenitor cells, stem cells, neurons, nematocyst precursors, and germ cells express genes from families that predate metazoans. Among differentiated cells, some express genes from families that date from the base of metazoans, like gland and neuronal cells, and others express genes from newer families, originating from the base of cnidaria or metazoa, like nematocysts. Interstitial cells contain translation factors with a function that has been conserved for at least 400 million years. I think that there is some health benefits in this animal. Thanks for watching. Remember to share.